Yesterday we looked at pure. We talked about pure. We talked about F1. We talked about F2. Slide your legs back just a smidge for me because I'll need to walk in front of you. So let's apply the purple and white that we did yesterday with our guinea pigs. When we said they were pure, what did that mean? Yeah, they were like self-fertilized, and they always came up, the trait that we were looking at always came up in their offspring, right? So if we had a black guinea pig and we crossed two black guinea pigs that were pure, then every single time we'd end up with all the offspring having that same trait, okay? Much easier to do in flowers because they can self-pollinate, right? A little bit more difficult, but at least the concept is there. The trait always passes through. So, if we use the letter B to represent the gene, we decided there were two alleles in there, right? Because, remember, the one was masked, the recessive one was masked, so Mendel decided there must have been two alleles. Then we could represent both alleles pure. Pure meaning they're the same letters. Now, why would have I made these capitals? Anybody remember that? How did we know they were dominant?
Because when they came out,
I'm glad y'all are all talking. That's a good sign. Let me say it again. So whenever we crossed two pure ones, all the offspring was all black, correct? That's right. So the allele, the dominant trait, ended up being black because brown was masked. So we could say then that our recessive, will be small letters. So dominant is represented by capital letters, uppercase letters. Recessive traits are going to be represented by lowercase letters. Does that make sense? All right. Is that something that y'all did in 6th grade? Did y'all put letters with them? All right. If you did Punnett squares, I'm going to assume you did. Terminology, genotype. Genotype. The word I want you to become familiar with, genotype. Notice the very first part, gene, genotype. Right here is how we represent the genotype. Right there is how we represent the genotype. So write the word down, genotype. And we are using genotypes, we're using letters to represent genotypes. So the alleles for black, alleles for brown. Now we said these are pure, correct? We said those are pure. Let's go over and look and see what our genotype ends up being after we cross these two. Hopefully this will work. I worked on this quite a bit today. All right, so we have two alleles, correct?
Yes.
Two alleles. We said that these are represented by two capital B's. So this is a capital B. This is a capital B. This is represented by small b's. When we cross these, what happens to these? In interphase, what happens to our chromosomes? Right, they replicate, right? They replicate. Then, once they go through meiosis 1 and they go through meiosis 2, what do we end up with? Four haploid cells. 4 gametes, or gametes. Same thing happens here. We started off, this is a diploid. There's both in the pair of homologs. In interphase, in S of interphase, they end up replicating, correct? In meiosis 1 and meiosis 2, we end up resulting in haploid cells that contain half as many chromosomes. Does that make sense?
Yes.
Now, in your book, did we represent 4 of these? They only represented 2. Any ideas why? No? What?
They didn't double them.
Maybe not smart enough. They didn't double them. Actually, it's because, and we're going to look down here in a second and see, these 2 will always end up the same, and these 2 will always end up the same. Why is that? Right. We just replicated these, right? So these 2 were replicated. These 2 were replicated. So they're just showing them. It's like they expect you to know by now that instead of showing the 2 replicated ones divided out, we're just going to show you what they are, what they would be originally. So we're going to look at that again. All right, so all of these haploid cells ended up with the dominant trait, correct? All of these ended up with the recessive trait. If this randomly fertilized with this one, this would be our resulting, right? And what would our genotype be for this? Yeah. So that's our genotype. Now, what if this one randomly met up with this one? Still the same, right? What if this one randomly met up with this one? Whoops. So no matter which ones, no matter if we randomly met up, we would end up with all the same, wouldn't we? And what all of them are is a dominant and a recessive. What would all the babies look like? If black is dominant, then all the babies would be black. Every single time we ended up through meiosis and we randomly went from egg to sperm, there is 100% chance that all of them are going to be what color? Black. 100% chance that they're all going to be black. Because in this case, we have a dominant allele. We have a dominant allele. All right. So we crossed these 2, and we said we ended up with all of our babies. Whoops. All of our babies ended up being black. Correct? Does that make sense? All right. Now, let's cross our F1 generation. So here is our F1 generation, and here is our genotype. What do we physically see? What color do we physically see? We physically see black. Our genotype, though, is a big B and a little b. So our genotype for our physical appearance is a big B and a little b, a dominant and a recessive. First thing that happens in interphase, in S phase of interphase, that's right, they end up replicating, right? These still code for coat coloring. They still code for coat coloring. They're homologous chromosomes. They both code for coat coloring. But this one codes it black. This allele codes for black. This allele codes for brown. Same thing happens here. They're going to replicate. Kind of hard to see there. Not a lot of control. All right, so we'll go through meiosis 1. Meiosis 1, we separated out homologous chromosomes, and meiosis 2, then we ended up separating out, and we have some left over in there. Now, let's see what ends up happening with our crosses now. So, possible sperm, possible egg. Don't know which one's right. Random fertilization. We never know which one of these is going to be the lucky sperm. We never know which one of those is going to end up being the lucky egg. Can you see okay? So if we have this one, whoops, we'll just move that out of the way since those are on the, all 5 million of them. Y'all know they're in a cell, okay? And this one, then what color would the baby be? Baby would be black. Now what if, because every single time a guy makes sperm or a girl makes an egg, we never know which one of these it's going to end up being. So what if we have this one the next time and then we have this one? Now we're going to have the option of a brown baby. So every single time we have another child, we get to start off with the same haploid cells. So every single time you have a child, you're going to have the same percentage of probability or chance of certain characteristics. We'll keep going. Yeah. Very good question. We'll lead into that. Yes, it depends upon if the trait's dominant or not. If the traits are dominant, no. If just one trait is dominant, no. And right now we're looking at complete dominance, but we're going to get to something that's called codominant, where they're both dominant. Mm-hmm. Exact same way. Yeah. Because where did they get the copies of these initial chromosomes? From their parents. And their parents. It depends on if it was dominant or recessive. I could have very easily said that instead of brown, we had white. And maybe white was our recessive color. So it just depends on if it's dominant or if it's recessive. Or like if Alex asked, maybe they're those dominant, and we'll look at those in a minute. Okay, got a question?
What is the type? Because they're both lowercase d.
We're going to get there. Yeah, we're going to get there. All right, so random, this one, random, this one. Black. Random this one, random this one. All right, no more brown ones, sorry. That one died. All right, so does that make sense so far? All right, so what would we say the genotype of this one would be? Back little b, little b. What's the genotype of this one? And if we have this one, and what color is it? Black. And it's black. Good. All right. So the genotype of homozygous dominant. Anytime you see the word dominant, you know it is going to be lowercase or uppercase letters. Uppercase. Dominant. Big letters are dominant. Homozygous. Homo meaning the same. So in this case, we would have homozygous dominant. Let's still use the letter B's. Homo meaning they're the same. Dominant would be both capital letters. Homozygous, so they're both going to be the same letter. But now we have recessive. So we're going to be lowercase, both the same letters. New term, Heterozygous. Write down the word heterozygous. So if we have homozygous meaning the same, heterozygous must mean different, one of each. So that's our genotype, what our genes would be, what each of the alleles would be. Phenotype, what we physically see. So now write down the word phenotype. This is the physical, what we physically see. If we have two big B's, black. Actually, I will just go ahead and, no, I won't. We would just know that would go there because I don't know if I can, oops, sorry. Maybe I can do order. Bring in front. Bet you it will. If I can keep it on there long enough. What'd you say? Now, homozygous recessive?
Brown.
Brown. Heterozygous, if this is dominant, then the dominant trait, this allele is going to be dominant over this one. So just like with the purple and the white flowers, the white allele is still there in that flower, but you don't see it. The brown allele is still here, but we don't physically see it. Does that make sense? All right, so now let's apply this to this.
How do you get a spotted animal?
That's codominant. We're going to talk about it in a second if I can ever. Let's just pull this up right here. Because apparently I can't slide. There we go. So, does anybody remember what this is called?
Karyotype.
Karyotype. Very good. It's a karyotype. A karyotype, this is a human. This is for a human because there are how many pairs of chromosomes? 23. And what are our X and Y chromosomes called? Sex chromosomes. Is this a male or a female?
Male.
Why?
Y.
Because there's a Y. That's right. What are the rest of all these other chromosomes called? Autosomes. Y'all are so incredibly smart. What do chromosomes code for? Traits. They're going to code for our traits. Specifically, they're going to code for like a guinea pig, or for us, they would code for hair color. So it would be like chromosome 3 right here. And this would be a section. These are homologous chromosomes. This would be a section of the nucleotides. Remember those nucleotides? Adenine, thymine, cytosine, guanine?
Yeah.
It would be a section of those nucleotides that are going to code for a protein that causes the hair to be black or to be brown. Oh, I'm just random. Yeah. All right. They do know, because of the Human Genome Project, they do know about 80% or so of, maybe even higher now, 90% or so of where all characteristics are coded in DNA. It was just 2 years ago that they determined where blue eyes were coded. So this is all new stuff, really still new stuff.
Okay, how is it when you're little then? Like your hair will change color.
Mhm.
Yeah, Yep.
So a karyotype is what can show us, it shows us a photograph of the genes, it shows us photographs of the genes, of the chromosomes, and we know that certain areas on homologous chromosomes are going to be coding for the same trait. Now, the majority of the time in humans, we don't have just one chromosome that codes for these traits. Normally they span across more than one chromosome. But at this level, we're only going to talk about them being on one chromosome or another chromosome because it starts getting really, really complicated, but very, very interesting. All right, so let's make a Punnett square on your paper. You're going to draw a square. Thank you. Quite proud of that. Does it matter if our homozygous dominant's here or our homozygous dominant's here?
No.
No, does not matter at all. Does not matter at all. So we're just going to randomly choose. Now, I want to show you on the first one or two what is going on here. So we have got that one, our homozygous dominant and our homozygous recessive. So we're going to separate out just like we did back here. How we ended up separating these out. This is basically making Punnett squares, just seeing what we might end up with. If this one ends up with that one or this one ends up with that one, that's what we do with Punnett squares. So we're going to separate these genes out. So we said that one has one, that one has one, this is brown. But we never know who's randomly going to match up with who, right? We never know who's going to randomly match up with who. So you're going to pull across. Let me do it this way. This is the first time I've done this way. So y'all remember how to do this? Can you fill out your Punnett squares? Do you remember how to do it?
Yeah.
I'll let you follow me. Whatever's written up here, we're going to write it all the way down. So we're just seeing if egg and sperm, if these homologs get together, what would the resulting baby end up being? Whatever's written up here, we're going to write all the way down. Whatever's written over here, we're going to write all the way across. Whatever's written here, we're going to write all the way across. That familiar? That look familiar? I want to show you what that means gene-wise. As long as I don't randomly write on stuff. So we would carry, just like we did a while ago, whenever we separated out our four So these would end up being our zygotes, what our possible zygotes would look like. What color would all of those end up being?
Black.
All of them would end up being black, right? So what did we do? We just crossed two pures, and we ended up with an F1 generation. So we just put in a Punnett square what we kept randomly doing over here. So it makes it a very neat and organized way to be able to see what are our possible outcomes. So when we look here, we have 4 possible outcomes. So we have a total of 4. And what is the fraction, or how many would we possibly have that would end up with the genotype heterozygous, big B, little b? 4. So there's a 4 out of 4 chance that they are all going to be what? Heterozygous. Or there is a 4 out of 4 chance that phenotype, they would all be black. So let's see what happens here. Now we're going to take and we're going to cross our F1. Our F1, we know that, I hate it when I do that. We know that they are both black, right? But they are carrying these genes, aren't they? So they're still carrying the brown genes. So let's go ahead and do your Punnett square. Big B, little b. Big B, little b, and everybody do your Punnett square. I know, isn't that unbelievable? Gotta feel my hands. crazy. Whatever you have written at top, you're going to carry it all the way down. Whatever you have written up top, we're going to carry it all the way down. Whatever you have written out to the side, we're going to carry it all the way across. Whatever you have written out to the side, we're going to carry it all the way across. Now, every single time you've got sperm or egg being made, they have the same percentage of matching up this way. So let's look and see what we have. Two big B's. What would we, so genotype is homozygous dominant. What would we see? We would see black. Big B, little b, heterozygous. What would we see since black is dominant? Little b is the only time we're able to see the brown recessive trait. There are a total of 4 possibilities. What is the possibility for our phenotype to be black? 3 out of 4. There's a possibility of 3 out of 4 that we're going to have black ones for phenotype. What's the possibility that we're going to have a brown one? 1 out of 4 opportunity every single time, right? Now, genotype. There's still a combination of 4. What is the possibility that it's going to be homozygous dominant? 1 out of 4 for homozygous dominant. What's the possibility it's going to be heterozygous? 2 out of 4 or 1/2. What's the possibility that's going to be homozygous recessive? 1 out of 4. All right, does that make sense? Good. All right, now, let's cross this. Let's cross I hate it when I do that. Let's cross a black one. One that is heterozygous, and a brown one. Better go ahead and cross those two. Let's see what we have. So we've been doing fractions. Now I'm going to show you another way to write what we see. Doesn't matter which one we put on top or which one we put on the bottom. For phenotypic ratio, go ahead and write out the word phenotypic ratio. For a phenotypic ratio, you normally do dominant to recessive. For a phenotypic ratio, we do dominant to recessive. So let's see phenotypically, what would we physically see? What would we physically see here if this one made black? The big B would dominate. We would see black here. If this one made, we'd see brown. And if that one made, we'd see brown. So phenotypic ratio would be 2 dominant to 2 recessive. 2 dominant to 2 recessive. And you could go ahead and reduce that and say it's a 1-to-1 ratio. Now, genotypic ratio. Genotypic ratio. In genotypic ratio, you normally end up going homozygous dominant to heterozygous to homozygous recessive. And I normally write it capital HD because homozygous dominant means 2 capitals. Heterozygous, capital H, small t. Heterozygous is 1 of each, lower and uppercase. And homozygous recessive, 2 lowercase. So let's count and see. And I want you to be able to remember this order. Homozygous dominant on one side, heterozygous on the other side. Homozygous recessive on the other side, heterozygous in the center. How many homozygous dominant do we have?
Yep.
How many heterozygous do we have?
Yep.
How many homozygous recessive? So that ratio would be 0 to 2.